T cells can be broadly broken down into two categories, naive and effector T cells. A naive T cell is a mature cell that has undergone differentiation in the thymus, expresses a T cell receptor, and circulates between the bloodstream and secondary lymphoid tissue in search of antigen specific to its particular TCR. The term naive reflects that the T cell has not yet encountered its specific antigen. When a naive T cell encounters antigen in the form of a peptide MHC complex, that T cell will be induced to clonally proliferate and differentiate into an effector T cell that can help remove its recognized antigen. It is important to note that these effector cells are limited and that they can only recognize antigen subsequently in the form of peptide MHC, expressed on host cells. For example, an effector T cell will not be able to attack a bacterium or virus if it is encountered natively in the bloodstream. If a virus has infected a host cell and that target cell displays viral protein via its MHC, then that specific effector T cell may elicit its cytotoxic effects. Recall that there are two classes of MHC, each with specificity to a certain population of T cells. MHC class II, which is exclusively expressed on antigen-presenting cells, is recognized by CD4 positive T cells. MHC class I, which is expressed on every host cell, is recognized by CD8 positive T cells. Naive T cells can differentiate into different types of effector cells, and their fate is largely determined by whether they expressed CD8 or CD4. CD8 positive T cells differentiate into cytotoxic T lymphocytes, or CTLs. When CTLs encounter antigen on a target cell via MHC class I, a swift and efficient cell-mediated killing reaction takes place via the membrane attack complex or FAST pathway. CD4 positive T cells can differentiate into a number of different effector cells, each with unique function. The cytokine milieu present at the time of priming significantly influences the fate of the CD4 cell. Four of the main CD4 subsets are the T helper 1, T helper 2, regulatory T, and Th17. They'll be discussed later. Time for a quiz. Take the next 20 seconds to answer the question above. In what tissue or social media platform are T cell responses first initiated? You got it. T cell responses are first initiated in peripheral lymphoid tissues, such as the lymph nodes or spleen, where naive T cells become primed through antigen exposure by antigen-presenting cells, such as dendritic cells. Over the next series of slides, we will illustrate how a T cell response is initiated, particularly in lymph nodes. Recall that blood and lymph are two separate compartments. Therefore, lymphocytes require a mechanism by which they can home to the lymph nodes. This process is dependent on adhesion molecules and chemokines so that the cell can enter the lymphoid tissue. High endothelial venules are specialized vessels expressing glycam-1 and CD34, both of which express the carbohydrate moiety sulfated siallo-Lewis X. L-selectin expressed on the lymphocyte binds the carbohydrate moiety, which forms a loose interaction that facilitates the lymphocyte rolling along the endothelial surface. Lymphocyte rolling brings the lymphocyte receptor, CCR7, into closer proximity with its ligand, CCL21. This interaction initiates a signal transduction cascade, causing a conformational change in LFA1, which allows it to bind with high affinity to endothelial ICAM1. At this point, the lymphocyte is tightly anchored to the endothelium and is able to migrate into the lymph node. As mentioned, naive T cells require initial antigen exposure by an antigen-presenting cell to differentiate into an effector cell. Dendritic cells are the most efficient APC and undergo a phenotypic switch from a phagocyte to an APC upon activation. In peripheral tissues, immature DCs display long dendritic processes and great surface area for encountering pathogens and antigens. Fluorescent staining of MHC class II in green and lysosomal protein in red displays a high degree of colocalization in endosomes at this stage. Immature DCs are very phagocytic in nature and can become activated by toll-like receptor stimulation, tissue damage, or inflammatory cytokines. Activated DCs leave the periphery and migrate to the nearest lymph node via lymphatics. At this stage, the DC is no longer phagocytosing antigen, MHC class II expression is visible on the cell surface, and the cell has undergone a morphological change with highly folded cell membrane yielding the classic veil appearance. Once in the lymph node, surface MHC class II expression is maximal for antigen presentation to the lymphocytes. On electron microscopy, a DC can be seen interacting with a T cell. If the antigen matches the TCR specificity and other co-stimulatory signals are present, the T cell may proliferate and differentiate into an effector. Dendritic cells can process and present antigen in various ways. Extracellular pathogens, antigens, and virus particles can be taken up by either receptor-mediated phagocytosis or micropinocytosis, where they ultimately are delivered to endosomes and loaded onto MHC class II. If an antigen makes it to the DC cytosol, whether it be by viral infection, cross-presentation, or direct transfer, the peptide will be loaded onto MHC class I. Moving on, next we will detail the molecular events that occur when a naive T cell is primed by an APC. The concentration of APCs in T cells is very high within the lymph node. In this tissue, the transient binding by various adhesion molecules allows the attached lymphocyte to sample peptide MHC present on the APC. If the antigen matches the TCR, a signaling cascade will trigger a conformational change in LFA1 that lengthens and strengthens the cell-cell interaction. The TCR-MHC interaction is required but not sufficient for the effective expansion and differentiation of the naive T cell. A co-stimulatory signal is required for a full response, and the best characterized interaction is between that of B7 and CD28. Signal 1 increases the transcription factors responsible for transcribing IL-2 RNA. Signal 2, in addition to increasing these same transcription factors and AKT, also stabilizes the 3' UTR of the IL-2 RNA, which enables each copy of RNA to be translated a greater number of times. The combination of signal 1 and 2 induce IL-2 and other factors to drive the cell into G1 and fosters a maximal proliferative response. Additionally, third signals from other interactions and cytokines can further influence the differentiation of the naive T cell into a particular effector cell. In addition to increasing IL-2 production by the T cell, activation also promotes production of the IL-2 receptor alpha subunit, also known as CD25. The 3-chain IL-2 receptor has markedly increased affinity for IL-2, which further enhances the cell's ability to respond to IL-2. So in summary, at baseline, naive resting T cells only express moderate affinity IL-2 receptor and have minimal IL-2 production. Upon activation, the T cell produces high affinity IL-2 receptor and secretes IL-2, which binds in an autocrine and paracrine fashion to accelerate cell cycling and clonal expansion. If you'd like, take the next few moments to answer the above question. T cells activated in this way can divide 2 or 3 times a day for several days, allowing one precursor cell to give rise to thousands of clonal progeny that all bear the same antigenic receptor. Still, other costimulatory molecules exist. Inducible T cell costimulator, or ICOS, binds a structural relative of B7 but results in secretion of IL-4 and interferon gamma rather than IL-2. ICOS is predominantly expressed on T cells within germinal centers, so their secreted products help facilitate B cell maturation and proliferation. CTLA-4 is an inhibitory protein that binds B7 with higher affinity than CD28. CTLA-4 is expressed on activated T cells to limit IL-2 production and ensure that proliferation is regulated. CD27 is another co-stimulatory receptor on T cells. CD40L and CD137 are two bidirectional stimulators that activate the DCs in T cells to increase responsiveness. OX40 is another stimulator receptor that has been implicated in sustained T cell reactions. An important distinction between naive and effector T cells is the lack of need for co-stimulation post-initial priming. The next time an effector encounters its TCR-specific antigen, it will be able to kill the target cell. True or false? All CD8 T cells differentiate into CD8 cytotoxic T cells. True. All CD8 T cells differentiate into CD8 cytotoxic T cells. Interestingly, naive CD8s require more co-stimulation to reach effector status than naive CD4s. This may be due to their profound capacity for destruction, but there are two ways by which a naive can become activated. 1. DCs are the best APCs and intrinsically have high co-stimulatory potential. A naive T cell interacting with a mature DC can accomplish this. Alternatively, a lesser APC could be coaxed into higher co-stimulatory expression with the help of a CD4. If the CD4 cell is stimulated by the APC, it can be induced to express CD40 ligand and IL-2. The IL-2 will act on the CD8 to increase proliferation, while the CD40-CD40 ligand interaction will upregulate the co-stimulatory markers B7 and 4,1-BBL on the APC to increase co-stimulation of the CD8. By this mechanism, the CD8 can achieve the co-stimulatory threshold necessary to activate and expand. In contrast, CD4s have multiple potential fates, and their differentiation pathway is largely cytokine-driven. As one might expect, different cells have different functions which are detailed here. T helper 1s classically aid in intracellular killing, such as by inducing oxidative burst genes. Th2s help recruit eosinophils, basophils, and mast cells to help promote barrier immunity and defense against parasites. Th17s promote innate immunity through the recruitment of neutrophils and induction of antimicrobial peptides by the epithelium. Follicular helper T cells are key for promoting B cell maturation within the germinal centers of B cell follicles. Depending on the cytokine secreted, follicular helpers can promote tailored antibody responses to a number of different pathogens. Regulatory T cells suppress immune activation and are important for preventing the development of autoimmunity. Helper and regulatory T cells elicit their function through the cytokines and surface proteins they express. Here is a chart highlighting the function that corresponds to each cell type and effector molecule. Similar to a naive T cell encountering an APC, the immunological synapse is first facilitated by nonspecific adhesion via LFA and CD2. If the TCR encounters specific antigen, then the cell-cell interaction will tighten and reorient the microtubule cytoskeleton so that granules containing effector molecules are polarized to the site of the cell-cell interaction. This is where they can be released in a precise manner towards the target cell. Release of cytotoxic granules by the CTL will induce apoptosis in a highly targeted and specific manner. You can visualize the stages of apoptosis by EM in the bottom panel. You can see a healthy cell to the left. The middle cell displays condensation of the chromatin and blubbing off of the membrane. In the third panel to the right, you see a highly pycnotic nucleus with a cell that has lost the majority of its membrane and cytosolic contents. After induction, a CTL can move to another target cell and induce the same process. Can you name the correct cytotoxic protein based on its target cell action? Very good.